The study of genetics mainly revolves around the study of genes, genetic variation and heredity in living beings. The late 19th century was the era of understanding of genes and genetics. Before going into the details of inheritance, we must know certain terms. Alleles are alternative forms of the same gene that are located at corresponding positions on homologous chromosomes. Alleles can be dominant or recessive. Genotype is the allele present in the DNA of every organism. A pair of letters is used to represent a genotype, like capital T, capital T, capital T small t and small t small t. There are always two letters in a genotype because one trait comes from the father and another comes from the mother. Alternative form of gene is allele. Allele can be dominant or recessive. If an individual has two of the same allele, whether it is dominant or recessive, it is called homozygous and represented as capital T or small t. On the other hand, an individual which has one each of two different alleles is called heterozygous and represented as capital T small t. Gregor John Mendel is called as the father of genetics. He studied trait inheritance, that is, the flow of patterns in which traits move from parents to offspring. He was the first person to observe that trait inheritance occur through a discrete unit of inheritance, which we call as the gene. He worked on Pisum sativum, also known as the common pea plant, and brought out the laws of inheritance. Although the principle of inheritance given by Mendel were on plants, they can also be employed on animals and human beings, because the transfer of trait is the same for all complex forms. Mendel worked on purebred pea plants which were developed through natural self-fertilization through many generations. A single alteration in a trait was therefore demonstrated by a visible difference between varieties. With the experiments, he reached various conclusions. Units or factors are unchanged when they are being passed on to the descendants. These are now referred to as genes. An offspring receives one such unit from each parent for each trait. A trait may not be visible in an individual. However, it can be passed on to the next generation. It is important to note that in his experiments, the starting parents were homozygous for seed color. That means they each had two identical alleles for each trait, two yellow or two green. On the other hand, the progeny in F1 generation were all heterozygous. In other words, they each had inherited two different alleles. A dihybrid cross is a cross between two purebred varieties that differ in their observed traits. For genes on the different chromosomes, each allele pair shows independent segregation. If F1 generation produces 4 identical offsprings, the F2 generation, which occurs by crossing F1 generation individuals, shows a phenotypic ratio of 9:3:3:1. Variation in characters may be governed by multiple genes. They are not only separate elements producing different individual effects, but can also interact to produce completely different phenotypes. Gene interactions may give modified F2 or second generations. This happens because characters are influenced by two or more gene pairs which interact with each other. In all his preliminary experiments, Mendel worked on traits with single gene control and that one allele was always dominant on the other. The rules that Mendel derived do not explain many inheritance patterns. One exception to his rule is that one allele is not always completely dominant on the other. There are intermediate phenotypes between two parents and there is no dominance. This pattern of inheritance is called as incomplete dominance. That means a cross between organisms with two different phenotypes produces offspring with a third phenotype, which is called blending of parental traits. For example, one gene of snapdragon flower has two alleles, one for the red flower and other for white flower. A plant that is homozygous for red allele will have a red flower, while another which is homozygous for white allele will have a white flower. On the other hand, the heterozygous plant having both the alleles will have pink flower. Neither the red nor the white allele is dominant. So, the phenotype will be a blend of both the parents. In humans, one of the best examples would be the hairs on a baby's head. If one of the parents has curly hairs and the other has straight hairs, the chances of the child having wavy hairs are the most. It is the intermediate between curly and straight hairs. Apart from genetic traits, there are certain diseases like Tay-Sachs disease and sickle cell anemia which are associated with incomplete dominance. Sickle cell anemia is disease in which hemoglobin protein is incorrectly produced and red blood cells have sickle shape. A homozygous recessive person will have abnormal shape of red blood cells, while a homozygous dominant person will have normal shape of red blood cells. Meanwhile, due to incomplete dominance pattern of this trait, a person who is heterozygous for sickle cell trait will have some abnormal cells and some normal cells. These heterozygous individuals have a fitness advantage. They are resistant to malaria parasite. In codominance, neither phenotype is recessive. Both the alleles are equally represented and neither dominates the other. Thus, two different alleles or phenotypes are expressed in heterozygous individuals. ABO blood group system is the best example of codominance. IA, IB and I are the three genes for blood types. I causes O type and is recessive to both A and B alleles. The A and B alleles are codominant with each other. The person having A and B allele has AB blood group type. Here A and B antigens are both expressed on the cells. This pattern of inheritance is completely different from the Mendel's rule because both alleles are expressed completely and one does not mask the other. Another example is the color of a flower. The pure line phenotypes of the flower consist of red and white color. When plants of parental red and white colored flowers were crossed, F1 generation produced pink flowers. On the other hand, in F2 generation, when F1 plants were self-crossed of the plants that were thus created, 1/4 produced red flowers, half of the plants produced pink flowers, while the remaining 1/4 plants produced white flowers. Phenotypes that are revealed by biochemical experiments are restriction fragment length polymorphism, RFLPs or DNA markers, isozymes or protein size markers, immunological reactions and quantity of metabolites. For example, suppose that a gene of interest resides on 3 kb and 2 kb DNA fragment of first and second parent respectively. When a cross occurs between the two parents, one chromosome carrying the gene comes from each parent. In F2 generation, since the technique recognizes both copies of parental DNA, the signal will be 2 times stronger than F1 generation signal because of one chromosome from each parent. However, F2 generation will segregate for 3 genotypes in a 1:2:1 ratio. In this, genotypic designation will be 3 kb fragment is allele A1, the genotype of parent 1 will be A1A1, and 2 kb DNA fragment is allele A2 and the parental genotype will be A2A2. F1 generation will be heterozygous having A1A2 genotypes, and F2 generation will be 1 A1A1 is to 2 A1A2 is to 1 A2A2. So, the major difference between incomplete dominance and codominance is that in incomplete dominance there is an expression of alleles that are neither dominant nor recessive. These alleles mix together and result in physical traits which are a mixture of both the alleles, while in codominance both the alleles exist in a simultaneous manner. That means, both the alleles are expressed in the progeny. Epistasis is a phenomenon where the effects of one gene are modified with one or more genes. These genes can be called as modifier genes. In incomplete dominance, there clearly is epistasis, whereas in codominance this may not always be present. That means, they are independent and coexist. Literal meaning of epi plus stasis is to sit on the top. It is an interaction of alleles at two different gene loci that affect a single trait. When the recessive alleles of one gene conceals the effects of either allele of the second gene, it is called recessive epistasis. The color of the coat of a Labrador dog is classical example of this type of effect. A golden color Labrador was crossed with a black colored Labrador. Then in F1 generation, all black colored Labradors were produced. When these F1 generation dogs were self crossed, 9 black, 3 brown and 4 golden Labradors were produced in the F2 generation. Pigment production B and subsequent incorporation E into the hair shafts are controlled by two separate genes. Black color is produced when both the genes function. Mutation in B leads to brown coat and mutation in E leads to absence of pigment. When the dominant allele of one gene masks the effects of either allele on the other gene, it is called dominant epistasis. For example, in a flower, one gene locus may determine white or green pigment. Alleles of a locus capital W and capital W small w produces white and small w produces green flowers. However, in the presence of a dominant allele capital Y at another locus, the flowers produce no pigment, resulting in white flowers. The allele capital Y is therefore epistatic to both capital W and small w combinations. Therefore, in a cross between capital W, capital Y, small y plants, 12 plants will bear white flowers, while 3 will bear yellow flowers and 1 will bear green flowers. Duplicate recessive gene occurs when two loci affect the same phenotype. If both the gene loci has homozygous recessive alleles and identical phenotypes are produced, then the ratio will be 9:7. For example, if pigment color is represented by capital C or capital C, but not small c, and by capital P, small p, but not small p, then pigment will be produced in any genotype except small c or small p. That means, both loci must have one dominant allele to produce a desired phenotype. The ratio of 9:7 for pigmented is to unpigmented will be achieved. When one dominant allele of both the genes loci produces the same phenotype without cumulative effects, it is known as duplicate dominant genes. An example is parental red and white flowers of a plant which explain the color of progeny flowers. White flower will have small a, small b genotype, while all other genotypes will be expressed as red flowers. The characteristic ratio is 15:1 with pigmented to unpigmented plants. In case of inhibitory genes, one gene when dominant is epistatic to other, but other recessive homozygous gene is epistatic to the first. Dominant recessive epistasis takes place. For example, feather color of fowls. Gene 1 inhibits production of feather color even when gene for color production is present. Another homozygous recessive gene interacts and prevents color production because of unavailability of the substrate for color production. This type of interaction is suppression gene interaction. Suppression of one gene by other gene can be demonstrated by production of malvidin chemical in plant genus Primula. Dominant gene K is responsible for pigment production. However, action of dominant K gene may be suppressed by non-allelic dominant gene D. When F1 dihybrid progenies were crossed, capital K, capital D, small d, they produced 13 malvidin minus is to 3 malvidin plus individuals in F2 generations. Polymeric effect can be explained when phenotypic expression depends on the dominant allele of two gene loci and the dominant one will be the phenotype. The characteristic ratio is 9:6:1. It is complete dominance of both gene pairs. However, when one gene is dominant, it hides the effect of another. For example, in summer squash fruit, gene pair A and B results in sphere shape which is dominant over long shape. The effect of these dominant alleles at two loci will be additive and produces dominant phenotype, either homozygous or heterozygous. F1 generation produces disc shaped fruits, and on self cross between disc shaped fruits 9 discs, 6 spheres and 1 long phenotype are produced. Today we have discussed various aspects of Mendelian genetics. The knowledge of classical genetics is of utmost importance to all biology students, even though today its practical importance seems to be related only to counseling in certain diseases. However, understanding of Mendelian genetics is the basis of modern genetics, which now rules the biological world.